Hi, let's understand apoptosis in detail. So, what exactly is apoptosis? First, we need to understand the definition of the apoptosis. Apoptosis is a genetically programmed ATP dependent type of cell death that is induced by a tightly regulated program in which the cell which is destined to die will activate some kind of intrinsic enzyme, and that intrinsic enzymes will lead to degradation of all the cellular and nuclear and cytoplasmic proteins of that cell. That we know apoptosis is a program and control cell death. The next questions comes here: why does the cell decide to commit suicide? What are the trigger for the apoptosis? So that is the causes of apoptosis. We learn about this. Unlike necrosis, which is always pathological, apoptosis can be physiological as well as pathological. Under what all condition physiological apoptosis can be seen? Like under what all normal condition we have the apoptosis. See, during development, for example, our fingers are initially connected by a web-like tissue. Apoptosis remove this tissue, separating the fingers. Okay, if apoptosis fails here during our embryogenesis, we will be having syndactyly, that webbed finger. Okay, during hormonal withdrawal, some cells survive only in the presence of hormones. We know that when hormone decreases, these cells undergo apoptosis. For example, endometrial cell during menstruation, breast gland cell after lactation, prostate cells after androgen withdrawal. These are the example of hormonal withdrawal. Up. This apoptosis is also important for the cell turnover. Cells in the tissue like skin or intestinal epithelium are continuously need to be replaced. Okay, the old cell dies by apoptosis while new cells are formed. Apoptosis is very important for the regulation or immune system. We need to eliminate the immature lymphocyte there only, the removal of self-reactive lymphocyte. So we need a immune regulation to regulate the immune system. We need apoptosis. Okay, in what all condition we can see pathological apoptosis? Then whenever cells have a severe DNA damage, accumulation of misfolded protein, the cells have some in kind of infection, viral infection and all, there comes the pathological apoptosis. Now see, what are the morphological changes we will see in the cell undergoing apoptosis? First, under electron microscopy, we can अच्छे से visualize all the morphological changes. The first change that we can see is the shrinkage in the cell size. Then when this is the normal cell, when it will be going under apoptosis, first the cell will shrink, it will become smaller in size. After that, chromatin will start condensing. This is the hallmark of apoptosis, chromatin condensation. And then it will form blebs like this Mickey Mouse, no? Like this, formation of cytoplasmic blebs, and lastly apoptotic body. These apoptotic body will be taken up by macrophages and it will undergo phagocytosis. We'll study about this phagocytosis also. Then what are apoptotic body? Apoptotic body are bounded by a thin plasma membrane. So the degraded contents inside the cell does not leaks out in the surrounding. The content will not leak out, so there is no inflammation in apoptosis. Understand the mechanism of apoptosis in detail. The hero of apoptosis is the caspases. What are the caspases? Caspases are cysteine protease, that enzyme that cleaves the protein after aspartic acid residue. But remember one important point: these caspases are produced as inactive enzyme, as a pro-caspases. They need to be activated first. Procaspases are need to be activated at caspases. Caspases are further as initiator caspases, executioner caspases. Initiator caspases receive the signal and activate the other caspases. So what are the initiator caspases? Caspase 9, that comes from intrinsic one, and caspase 8, that comes from the extrinsic pathway. These intrinsic caspases will activate executioner caspases once these executioner caspases get activated, they lead fragmentation activation of cells. They break down the nuclear membrane, destroy the cytoskeleton of the protein, causes bleeding that we have seen in the morphological features, no, so that the cell dies in an organized manner. As apoptosis happens in a two main phases, initiation phase and execution phase. The initiation phase, the cell receives the sense of a death signal and activate the initiator caspases that are 8 and 9. Under executional phase, the executional caspases 3, 6, 7 break down the cells into apoptotic body, which is further removed by the macrophages and phagocytosis happens. Apoptosis occurs in two major phases, that is initiation phase and execution phase. In the initiation phase inside, the cell receive the death signal and decide to undergo apoptosis. From where does this death signal comes from? These death signals come from two different pathway, that is mitochondrial pathway and death receptor pathway. Mitochondrial pathway is also known as intrinsic pathway because the signal is coming from inside the cell, and in death receptor pathway the signal is coming from outside, that's why extrinsic pathway. Now let's understand the mitochondrial pathway. Remember, whenever the cell receives some kind of death signal, p53 plays an important role. When the cell repair is not possible, the BH3 protein get activated, and in turn the BH3 beam bad puma noxa, the bad guys, will activate backs and back will create a pore in the mitochondria through which cytochromes C start leaking out. When the cytochrome C comes out, it combine with other pro-apoptotic protein along with ATP and APAF1. This is the very important factor, APAF1. It form a apoptosome. This apoptosome lead to activation of caspase 9, and after that caspase 9 activate the executional caspases that are 3, 6, 7, and this again will play a role in nuclear fragmentation and breakdown of the protein. This was about the intrinsic pathway. We will understand about the extrinsic pathway. Signals come from outside the cell, so extrinsic pathway, the cell surface contain a special receptor that are death receptor. The most important death receptors are FAS CD95 tumor necrosis factor receptors. Now, when the fast ligand, these are the fast ligand, when the fast ligand bind to the fast receptor, trimerization of the death domain happen. When this trimerization happen, here comes the role of FADD protein, FAD protein. This FAS associated death domain, FADD is FAS associated death domain, will in turn recruit procaspase 8. This procaspase 8 is in inactive form. This procaspase will get activated as caspase 8. Okay, what is this? Some viruses produce a protein called flip. Flip block the activation of caspase 8. So flip prevent the apoptosis so that virus can remain inside the infected cell. This was the role of this flip protein which comes from viruses. Remember, extrinsic E, and E for death and E for it. The caspase 8 is coming from extrinsic pathway. Mitochondria. Mito is mine is 9, that is 9 caspases. Caspase 9 is from intrinsic pathway, caspase 8 is from extrinsic pathway. Now executional phase. Under executional phase, caspase 3, 6, 7 play an important role in breaking down the cytoskeleton protein, causing blame formation of apoptotic body. Apoptotic bodies. What are these apoptotic bodies? Apoptotic bodies are bounded by a plasma membrane, and inside the apoptotic body degenerated cells proteins and cytoplasmic organelles will be there. But because of this intact plasma membrane, the content do not leak out in the surrounding, and so no inflammation. So no inflammation in the apoptosis. How does this apoptotic body are taken by the phagocytosis? What are the process of phagocytosis of the apoptotic body? Apoptotic body will have some produce some apoptotic body will produce some kind of signal, that is eat me signal. We'll study about it. Remember, in a healthy cell, the phosphatidylserine present inside the plasma membrane, but in apoptotic cell, it flips outside. It flips outside, so it will express some kind of signal, and that is recognized by the macrophages. How the signals are produced? Signals are produced with the help of complements like C1q, and it will be recognized by the macrophage. This signal is known as eat me signal. Okay, and the process of phagocytosis of this apoptotic cell is known as efferocytosis. This was the mechanism of phagocytosis of the apoptotic cell. Now we'll revise the apoptosis in a concise manner. Okay, remember, apoptosis is a programmed cell death. It needs lots of energy. There is no inflammation. We have remembered that, we have studied that. Now coming to the pathway, mitochondrial pathway and death receptor pathway. Mitochondrial MI, that is intrinsic pathway, death DE like extrinsic pathway. Caspases are the hero of apoptosis. How many type of caspases? Initiator caspases and executioner caspases. What all are initiator caspases? Caspases 9 and 8 are initiator caspases, and these initiator caspases will activate the executioner caspases. Executioner caspases are 3, 6, 7. They cleave the key cellular protein of the cell. The major substrate of executioner caspases are PARP, DNA repair enzyme, nuclear lamine, cytoskeletal protein. These all, they will try to die the cell in organized way. They make the cell to die in an organized way. The executioner caspases. What are the process of mitochondrial pathway? The flow chart. Mitochondria become permeable because of that backs and back. Okay, and mitochondrial become permeable through which cytochrome C leaks. It will combine with ATP and APAF1. Formation of apoptosome will be there. Apoptosome activate the initiator caspase 9, and this caspase 9 will activate executioner caspase. That leads to execution phase. In execution phase, first the cell size shrinkage, chromatin condensation, nuclear blebbing, apoptotic body formation, and phagocytosis further will happen. Now coming to the death receptor pathway, here the fast ligand tumor necrosis factor on the present on the cell surface membrane. Here they will, when the receptor bind, trimerization of the domains happen, and FADD proteins play an important role in activation of initiator caspase, that is procaspase 8 of the extrinsic pathway. Here the caspase 8 will activate the executioner caspase 3, 6, 7. Again the same cycle, same phase will begin. Same thing. What are the key feature of the apoptosis? No inflammation, no leakage of cellular content, and it can be physiological as well as pathological. What are the clinical significance we need to remember? If we have decreased apoptosis, then also it will lead to disease. The cells are not dying. It will survive. The damaged cell will survive, leading to the cancer. Autoimmune disease, chronic infection will be there. That kharab cell will be inside the body. It will lead to chronic infection. Okay, if the apoptosis process is increased. Okay, in case of increased apoptosis, the cells are dying too early. Cells are dying, keep on dying. This will lead to neurodegenerative disease, Alzheimer's disease, Parkinson disease, ischemic injury, MI, stroke, like loss of T cells in AIDS. These are the where situations where there is an increased apoptosis. In what a physiological condition apoptosis during embryo development, we have the space between our finger, tissue homeostasis, elimination of damaged and aged cells, regulation of immune system. Remember, it's a very simple. So Let's revise. An intrinsic pathway inside the cell. Caspase 9 is the caspase of intrinsic. Extrinsic outside. Caspase 8. E for 8. Educational caspases 367. 367. Remember like this. BCL2 bodyguard of the cell. Anti-apoptotic. This want to live long. Anti-apoptotic. Backs and back make the pore in the mitochondria through which cytochrome C leaks out. BS3 protein, beam bad, puma noxa, these are the alarm protein. P53 quality control judge, it will only judge it. The cell can be repaired or not. If not repair, it will undergo apoptosis. Phosphatidylserine flipping out will produce eat me signal and then phagocytosis happen. IAP where the break, smack and Diablo where the break broom mover. Now, we'll try to revise again in a flowchart manner. First, DNA damage, growth factor withdrawal, endoplasmic reticulum stress will be there. P53 will come and check. It can be repaired or a repair is impossible. If the repair is impossible, then BS3 protein beam bad puma noxa will get activated. Backs and back will create a pore in the mitochondria. Through the pore, cytochrome C will be released. The cytochrome C will bind to the APAF1 and ATP formation of apoptosome. This apoptosome in turn will activate Caspases 9. Caspase 9 will lead to activate executional Caspases, and these executional Caspases will lead DNA fragmentation, cell shrinkage and formation of apoptotic body. Due to phosphatidylserine on the outer surface of the apoptotic body, eat me signal will be released and macrophages will recognize the signal. Apoptosis will take place, but nothing will come out, so no inflammation in apoptosis. Intrinsic one. Now coming to the extrinsic pathway. Here the cell surface receptor of fast ligand tumor necrosis, they will bind to the receptor fast CD95 and the formation of fat, and this fat will activate caspase 8. In turn the caspase 8 will activate caspase 367 and again it will lead to apoptosis. I think this makes the apoptosis very easy. So thanks, and try revising this flowchart with yourself for the exam and viva. Thank you.